Chronic lymphocytic leukemia in African ancestry population is characterized by increased telomere erosions. Hi, I'm Dr. Brian Koffman, the co-founder, chief medical officer, and executive vice president of the nonprofit CLL Society. The annual meeting of the American Society of Hematology, or ASH, showcases the latest breakthroughs in hematology. And I'm going to share through these monologues some of the abstracts that seemed important to me that you understand. I'm going to start with the bottom line. This abstract showed that this abstract reminded us that telomere length has been recognized as a significant and independent prognostic factor in multiple cancers. Shorter telomere lengths were found in Americans of African ancestry with CLL compared to those with Americans with CLL of European ancestries, even after accounting for other known prognostic factors. It's important to know who performed the research and where it presented, and as I stated, this was at ASH 2022, and it was a consortium of researchers across the US led by a group from Mayo that presented the findings. So, in the way of background, for those who don't know, telomeres are repeated sequences of DNA found at the very end of all our chromosomes. The telomere's role is to protect those ends of those chromosomes. Each time a cell divides, the telomere becomes slightly shorter. Eventually, they become too short so that the cells can no longer divide successfully, and this leads to cell death. In CLL, telomere shortening can lead to a loss of function of DNA damage control, the checkpoint genes. This in turn allows further cell proliferation and telomere shortening, sort of a feedback loop. What was the method? DNA was collected from CLL cells and used to measure the average telomere length through a method called quantitative PCR, polymerase chain reaction. 5 CLL samples with known telomere lengths were included in every section of research, every piece of telomere that was looked at in order to account for variability. What were the results? Telomere erosion, which is a fancy way of saying shortened telomeres, was evaluated in a total of 133 Americans of African descent with a mean age of almost 63, and 147 age-matched Americans of European descent with an average age of 62. The African Americans samples were 71% from males, and a similar 71% had unmutated IGHV, which confers a worse prognosis. The European American samples were 77% male patients and 66% unmutated IGHV. So similar comparator groups. As expected, the researchers observed shorter telomere lengths in the unmutated IGHV CLL compared to the mutated immunoglobulin heavy chain variable region IGVH in those where it was mutated. This goes along with the findings that the unmutated IGVH patients tend to have a more aggressive disease, suggesting that shortened telomere length is consistent with that. African American CLL patients presented with shorter telomere lengths when compared to the Europeans. They highlight that the difference in telomere lengths observed were restricted to the CLL cells. In other words, the way they looked for those CLL cells was to look for markers on those cell surfaces called CD5 and CD19, and if they were positive for that, that meant they were the CLL cells. And there was no difference in telomere lengths in the germ lines or the rest of all the cells in the African Americans versus the European descent Americans. So this was only happening in the cancerous CLL cells. When they stratified by the IGHV mutation, shorter telomere lengths were observed in the unmutated IGVH compared to the mutated IGVH in both cohorts, both the African Americans and the Europeans, respectively. The significantly shorter telomere lengths in African Americans remains significant even after accounting for all the other potential factors you think of, like their IGVH status, their age, their gender, the number of mutated CLL gene drivers, and their rise stage in multivariate analysis. So what's our conclusion? These results demonstrate that increased telomere erosion, or shorter telomere lengths, in Americans of African descent with CLL, when this is compared to those of European descent, it persists even after accounting for other known prognostic factors. At the same ASH conference, another paper, Racial, Ethnic, Sex, and Income Disparities in Overall Survival in Chronic Lymphocytic Leukemia Patients Seen in the Era of Targeted Therapies, Surveillance Epidemiology and End Results, or SEERS Registry Analysis 2009-2019, demonstrated poor overall survival in non-Hispanic Black Americans. Could the findings from this trial about telomere length contribute to those worse clinical outcomes? More research is needed. We include links to all of these trials on the website. Thank you for listening.